Welcome back. We are about to start part 2 of chapter 2, chemistry. So we're going to talk a little bit about chemical bonds and molecular structure, the structure of molecules. So the definition of a molecule is written here. You can see two or more atoms that are bonded together. And a molecular formula tells us which elements or chemical symbols of elements are found in a molecule. And if a element symbol is followed by a subscript, that subscript tells you how many of those atoms are actually in that molecule. When a molecule is made up of two or more elements, it is referred to as a compound. So there are three types of chemical bonds that are the most common. There's a fourth one that we will mention that's not listed here, but we will talk about it. So we have covalent bonds. Covalent bonds, there are two types, polar and nonpolar. We'll talk about hydrogen bonds and ionic bonds. The fourth that is worth mentioning is called van der Waals reactions. It's controversial. Sometimes it's considered a type of chemical bond. Others consider it just a very weak interaction. Van der Waals reactions are very similar to hydrogen bonds, but I like to mention them because they do play some roles in biology. So let's move into our types of chemical bonding. So covalent bonds occur when two atoms share electrons. Remember, we talked about in atomic structure that those valence shells, those outer energy shells in which some of the orbitals in those shells are not completely paired. They only have one electron instead of two. Those orbitals want two electrons. So many atoms will, in close contact, will actually share their electron, a single electron, such as between two nitrogen atoms or between two oxygen atoms. They will share their valence electrons in order to at least partially fulfill those orbitals or those outer shells. Now, this sharing of electrons can occur between two atoms that have the same atomic number. So we could have two of the same atoms, such as oxygen sharing with another oxygen, so O2, the oxygen that we breathe, or atmospheric nitrogen, N2. We could have two carbon atoms, so two atoms that have what we call the same electronegativity or attraction of electrons. Sometimes covalent bonds are formed between two atoms that are unequal in their electronegativity, so that one atom has the electron a little more often than the other. And so this is known as unequal sharing. And that is the difference between a polar and non-polar covalent bond. So we have this sharing of electrons, and some have a greater affinity than others. So if one of them has a greater pull or attraction, what we refer to as electronegativity for an electron, that electron will spend more time with that atom. So here in this example, we're just looking at equal sharing. So when we have electrons that are being shared between elements of very similar or equal electronegativity, that would be a covalent bond. Now here's the octet rule. We referred to this actually in lecture 1, but just want to go back and refer to how these atoms are shared, because these outer energy shells really want to fulfill that octet rule. They really want, each energy shell wants 8 electrons. So for many atoms, that valence shell will fill with 8 electrons when it actually starts to share with another atom. So here we have hydrogen and fluorine. Now hydrogen is actually an exception, one, because its valence shell is full with 2 electrons, so it can't meet that octet rule. But hydrogen can fulfill that rule for many atoms that need multiple electrons. So we can have oxygen wants 2 electrons, so it binds with 2 hydrogen atoms, and that gives us H2O, or water. A bond is considered a polar covalent bond when we have that unequal sharing. When we have two atoms of different electronegativities, they're going to share an electron, but because one of them has a stronger pull than the other, then the electron is going to spend more time with that atom. This creates a polarity or a charge difference across the molecule in its entirety, so that one side of the molecule, Let's take water, for example, since it's a very polar molecule. In water, oxygen has a really strong electronegativity, whereas hydrogen obviously does not. Doesn't have nearly as many protons and neutrons, only has one electron. So oxygen has this really high affinity for electrons. And when it shares 2 electrons with 2 hydrogen atoms, those electrons are going to spend more time with oxygen than they are with the hydrogen atoms from whence they came. So oxygen, which normally has a neutral, has no charge, will now have a slightly negative charge because it has these 2 extra electrons that have negative charges, so it's going to be slightly negative. The 2 hydrogen atoms would normally be neutral, but since this electron is further away and spending more time with oxygen, these 2 hydrogen atoms will now have a slightly positive charge. So this gives us a polarity or a charge difference across the molecule. Non-polar covalent bonds are simply where the electrons being shared, it's equal. So this would be usually between 2 atoms of the same element. There's no charge or polarity across the molecule. Now this plays a really big role in the characteristics of compounds made up of atoms that are, or molecules that are polar versus molecules that are non-polar. Non-polar molecules would include things like oils and petroleum type compounds. Polar molecules would be dissolved in water or something in water, sugar water. Think about salad dressing. Oil and vinegar do not match. Vinegar is a water-based or polar type molecule, and oil is not. So the 2 of them do not react. Think about it. Non-polar molecules have no charge. There's nowhere they have equal sharing of these electrons. There's nowhere for any kind of interaction to occur. There's no charge on that molecule. So there's no reason for that molecule to interact with others. Now, water is our, of course, textbook example of a polar covalent bond and a polar molecule. Electrons are more electronegative, so you can see here in red we have our oxygen atom, and then we have our 2 hydrogen atoms here, and here's hydrogen, and these guys only have these single electrons. These electrons are going to spend more time in the energy shell of oxygen. This is going to leave this kind of slight positive charge over here, right? We have this partial positive charge on the hydrogen atom, and you can see here we have a slight negative charge on the oxygen atom. And this gives this polarity or polar difference. We're positive on this side of the atom, but we're negative on this side. So as we move across, we end up with a polarity. From here we move on to hydrogen bonds, and hydrogen bonds are really just very, very weak type of covalent bonds. In the Lewis-Dash models, we see these dashed or dotted lines. That's representing a type of hydrogen bond. So you can see here they have these kind of dotted lines. This is a very strong covalent bond, so it's considered that polar bond. But when you see these dotted lines, it usually indicates some type of hydrogen bond. Now, individual hydrogen bonds are very, very weak. Okay? They're very weak. But if we have millions and millions and millions and millions of hydrogen bonds all together, then those bonds are very strong. The example I like to use is diving into a pool. If you are up on the high dive and you dive into the pool, you are going to, let's say you dive in feet first. You're going to point your toes. You're going to make yourself as slim and as narrow and as pointy as possible so that when you hit the water, you just glide right into the water. And you are doing that because water as a liquid is held together by, these water molecules are all held together by hydrogen bonds. Okay? Remember, we have that slight positive charge on the hydrogen side of the molecule and that slight negative charge on the oxygen side. And hydrogen bonds will form, I'll go back here, will form between this slight positive side here of this water molecule and the slight negative charge of the neighboring water molecule here. And because these are such weak charge differences, remember in love and chemistry opposites attract, so because these two charges are so weak, individually these bonds are very weak. So if we were diving into a pool of water and the liquid water is held together by hydrogen bonds between all of the water molecules, the fewer hydrogen bonds we have to break to get into the water, the easier and smoother it's going to be. So we just break very few, and we break very few at a time, and that allows us, with our pointed toes and our arrow-like structure, to glide very easily into the water. We're breaking as few hydrogen bonds as possible, and that allows us to glide into the water. Now, if I want to see how collectively millions and millions and millions of hydrogen bonds are actually very strong, I would do the exact same dive, but instead of making myself into the shape of an arrow and very pointy, I would do a belly flop. And a belly flop, you kind of land on top of the water and then sink down into it. And it's painful, and it's kind of like hitting concrete, and much more difficult to do because you are breaking millions of hydrogen bonds as opposed to just very few. So the more hydrogen bonds you have to break, the stronger it's going to be, or the more difficult it is to break it. This is why we can float some things on top of water that have a really large surface area, but if we take something that's that doesn't have nearly as much surface area, we can actually we can sink it. And a good way to take a look at this at home is get yourself a dish of water, or a glass of water. You can balance a penny flatwise on the surface of the water using the surface tension created by all of the hydrogen bonds in the water molecules. But if you take that same coin and you turn it sideways and try to put it on the water like you would be putting it into a coin slot in a machine, it'll sink right down to the bottom, and you won't be able to balance it on top of the water. So these hydrogen bonds individually, again, are very weak, but collectively they're very strong. This property of hydrogen bonds will become very important when we get into the structure of DNA. It is what actually holds the base pairs together in a DNA strand, and is very important in the strength and stability of genetic material. You can see here down at the bottom, individually weak bonds form and can break very easily when we move into the chapter on metabolism and we talk about protein function. When a substrate binds to an enzyme, hydrogen bonds are very often what hold them together, and we need them to have enough pull to bring the substrate and enzyme together, but be weak enough that when the enzyme is finished with its job, it can easily let go of the new product. So we'll go more into that in the protein structure. So here we have some water molecules being held together, and here are these dashed or dotted lines that indicate how these molecules are being held together. And you notice a hydrogen is always going to be attracted to an oxygen. So those are your hydrogen bonds in water. Here we're just depicting the hydrogen bonds that are holding together the bases, the internal bases in a DNA molecule. The sugar phosphate backbone, this red ribbon here, is actually held together with a form of covalent bond known as a phosphodiester. Now, ionic bonds are held together between two ions. We have cations and anions. Cations have a positive charge and anions have a negative charge. And when we have a cation attracted to an anion, we form an ionic bond. So an ion itself is an atom or a molecule, but usually it's an atom that has gained or lost an electron. So we have in its normal neutral state, one of the electrons gets lost to something else. We see this a lot in redox, what are called redox reactions, reduction and oxidation reactions in the cell. And when that molecule loses an electron, gets oxidized, or gains an electron, is reduced, this changes the charge of the molecule itself. And these molecules can then interact with each other based on charge. So always remember, in love and chemistry, opposites attract. So ionic bonds occur when a cation bonds to an anion, and the classic example of that is table salt or sodium chloride. Sodium and chloride each have a charge difference. So here we have a sodium atom right here. This sodium atom ends up losing an electron in some process, and so it gets oxidized. And over here, the chloride atom gained the electron, maybe it took the electron, and that leaves these two here, right? You notice our energy cells, this guy's energy shell is now full, right? And this guy's energy shell is also now full. It had that extra electron. And in that redox reaction, it pulled these two anions together. We have our cation over here and our anion over here. Pulled these two together to create an ionic bond. And so these two atoms form sodium chloride. And it's interesting because individually, chloride atoms are actually quite dangerous and can be explosive. Same with sodium, is highly reactive. But when we put them together in an ionic form, they become this inert table salt, this crystal salt that we can actually consume and makes our food pretty tasty. Chemical reactions themselves will occur when one or more atoms or molecules or substances, they get changed into others. And so we go from a reactant to a product. You're used to that from chemistry. And chemical reactions, all different types of chemical reactions, do share a couple of different properties. All of them will require some source of energy, usually to move forward. Reactions in living organisms oftentimes require a catalyst, and we call those catalysts enzymes. They're a class of they're called biological catalysts, and they're a class of protein. Most chemical reactions will occur in one direction, from left to right, from reactant to product. But oftentimes, particularly in biochemistry or biochemical reactions, we can very often see reactions reversing and moving from right to left, going from reactants to products, and then those products turning around and becoming reactants again. And when this occurs equally, we have the same number of reactants moving forward as we do products moving backwards. Of course, that's known as equilibrium. Most biochemical reactions are going to occur in some form of liquid environment. Water is a very big component in chemical reactions and does play a couple of different roles. So that means we need to talk about water and some properties of water. When a solution is formed, it has both a solvent and a solute. A solvent is what the solute is being dissolved in. So for most biochemical reactions, the solvent is going to be water. A solvent is usually a liquid. Most solvents, or water, is kind of what's considered the universal solvent. An aqueous solution, when you hear the term or you see the term in an aqueous solution, that always tells you that water was the actual solvent. The sugar was in an aqueous solution, or 10% aqueous glucose. That just means that glucose was dissolved in a 10% concentration in water. Ions and molecules that have polar bonds, so polar covalent bonds, are going to dissolve in water. Non-polar compounds will not. Water itself is a polar compound. So in this case, in terms of water, and when we talk about solutions, like with like. So oils, like oil, will dissolve in oils, polar in polar, non-polar in non-polar. How do these different compounds react with water? Well, we have hydrophilic and hydrophobic. A hydrophilic molecule is one that loves water, dissolves easily, is usually polar, and it's made up of polar covalent bonds. Hydrophobic compounds are water-faring. They do not like water and will actually be repelled by it, so they don't dissolve in it. And these are non-polar molecules, such as a hydrocarbon, which would be a carbon bound to some hydrogen atoms. An example of a hydrophilic compound would be when you make lemonade and you dissolve lemon juice and sugar in water. Both of those are polar compounds. Hydrophobic would be, like we said earlier, oil and vinegar. Vinegar is a water-based compound. Olive oil is not. So they don't mix. You have to shake them up. And when you shake them up enough to make the molecules of olive oil small enough, you get what's called an emulsion. An emulsion is not dissolved. It's just simply a vigorously mixed and broken up polar compound inside of a non-polar, or vice versa. Then we have what are known as amphipathic molecules. An amphipathic molecule is a molecule that has a charge difference across it. Now we're going to learn about the plasma membrane in this class, and we're going to be talking about what are called phospholipids. And phospholipids are kind of the most famous and most common amphipathic molecule. Phospholipids are used to make up our cell membranes. So amphipathic molecules are usually fairly large, and one side of the molecule has a polar or hydrophobic region, and then I'm sorry, hydrophilic region, and then we have the other end of the molecule will have a non-polar region. So just to show you real quick, here's a phospholipid, right? Here's the phosphorus glycerol molecule, and it has these two hydrocarbon tails on it. Now we know that hydrocarbons are non-polar, right? So this end of the molecule is non, writing with a mouse here. And then this end right here, this is a glycerol molecule, which does readily dissolve in water. So this part is polar. So one end of the molecule is nonpolar and the other end of the molecule is polar. So we have two different types of kind of molecule in one. Here's an example of hydrophobic versus hydrophilic. This is actually taken from a marketing campaign for a what's called it was, I don't know, sun something glass cleaner. It was a product that you would wash your windows on your house and then apply this product. It's kind of like Rain-X for your house. And the idea is that windows that had this traditional glass were kind of hydrophobic, and they would this water would repel, but it would trap all of this dirt. Right? The glass itself had a hydrophobic property, and it would end up trapping dirt, and you would get those water marks and dirt marks on your windows. And you would have to go out all the time, 3 or 4 times a year, and you have to wash your windows, and nobody likes to have to wash windows. So on the right over here, this sun clean glass was glass that they created that had a hydrophilic property to it, so that instead of the water sticking to the glass, the water molecules would actually stick to each other and run down the glass, taking the dirt with it, so that you didn't have to wash your windows. That was the idea behind it. Here's some other examples of some amphipathic molecules. Here's that phospholipid molecule I was telling you about. Here's how a cell membrane is formed. It's really kind of cool. The internal portion of the membrane here, the internal portion is made up of these hydrophobic tails. So they're attracted to each other. The cell membrane is actually bilayer. There's 2 layers. And then the outside portion here are the hydrophilic head parts, and that's what water can interact with. So this makes our cell membranes fairly impermeable to most molecules, because we have both a polar and non-polar region within the single membrane. Over here on the right, this is what's called a mycele. A mycele is formed by a detergent. Think about dish soap. Dish soap works really well against grease that we try to break down, because it has this non-polar region on the inside and then the polar region on the outside. So grease molecules, those non-polar oily molecules, get trapped inside of this region here, the inside of the mycele, and then we can rinse them away. Now, some detergents have greater ability to form these myceles than others, and that's why some brands of dish detergents you may like better than other brands of dish detergents. Solutions. Well, solutions are basically a compound dissolved in some type of solvent. So the amount of a solute that's dissolved in a single predetermined unit volume of solution is considered the concentration. And an example here is if we took 1 gram of salt and we dissolved it in a liter of water, this would be described as NaCl in a concentration of 1 gram per liter, right? And G per L. Molarity is how many moles. Remember that Avogadro's number from lecture 1. Molarity is about how many moles, or how many actual molecules, are in a particular predetermined volume. So the number of moles, and most of the time it's going to be in 1 liter, so it's moles per liter. Now, water itself occurs in 3 different states. It can occur as a solid, it can occur as a liquid, and of course as a gas. It changes in state. It can change from a solid to a liquid to a gas. I'm sure everyone is pretty aware of that. But this change from one state to another is going to involve some kind of input or release of energy. An input of energy would take ice to liquid, right? So a solid to a liquid, or a liquid and even more energy input would turn the liquid into a gas. And then, of course, as gas is cooled, condensation occurs, and the water that's in gaseous form would now become a liquid form. And if we pull even more energy out of those molecules, then it would eventually, we could freeze it and it would be ice. But water is universal, and it's mainly because it's so stable in its liquid form and its ability to dissolve things. Now, colligative properties are properties of a solution that depend on how many solutes are in a volume, not the actual solute itself. So colligative property of water just simply mean when sugar is dissolved in water, what properties does that water have? And it'll have different properties depending on how much of sugar is dissolved in it. That's all colligative means. So colligative properties will depend strictly on the concentration of particles, not on the actual type of particles. Think of sugar water. If we put just a little bit of sugar in water, the water is still very liquid-like, has a very low viscosity. But if I make something like a simple syrup where I put equal parts of sugar and water together and dissolve equal parts sugar in equal parts of water, then my water becomes very viscous and very thick. So it doesn't really matter what sugar is being dissolved in it, it's just how much of it is being dissolved. When we add solutes to water, we lower its freezing point. But at the same time, if we have to put energy in, it's going to raise its boiling point. Salt is supposed to do the opposite, but actually it does increase the boiling point of water, and this is why we add it when we cook pasta, because it helps to cook our pasta more quickly because the water is hotter. Sometimes animals will produce biological molecules that work like antifreeze. This prevents their body fluids, the water in their cells, from dissolving when they are in extremely cold environments. There are certain lizards and frogs that will do this. They'll bury themselves in mud at the bottom of a pond in the wintertime and go dormant and release these antifreeze-like molecules, because if cells freeze, they will bust open. Remember, water expands when it freezes, and this will cause the cells to break open and die. That's why we can't free our fingers and toes if we're in the cold. We can get frostbite. And frostbite is when our hands, the tips of your fingers, get so cold that the cells actually freeze and die, and then that tissue needs to be removed. Here's some examples: the emerald rock cod in Antarctica. Here, these are larvae, wasp larvae that release these compounds. These larvae hatch just before the wintertime in the northeast, in the far north up in Canada, and they will be able to survive over the winter without freezing and then become an adult wasp in the spring. But water has lots of other functions. It's not just used as a solvent. We sweat, right? We use water in our evaporation process. We use water to filter out waste products through our urinary tract system and our kidney system. Water is used for growth, the cohesive properties of water. Water has what's called cohesion and adhesion. In cohesion, water molecules stick to each other. Think of that hydrogen bonding that occurs in liquid water. And plants, look at this tree over here. This tree needs to get water all the way up, right? It's got to get water all the way up there. So the cohesive properties of water, as water moves up this tree, it's not a pump system, it's cohesion. The water molecules will use adhesion to stick to the plant tissue. Adhesion is water molecules sticking to a substrate. So as the water molecules are attracted to the actual tissue in the plant that pulls the water up, they will bring with them, they will attract behind them other water molecules, so that all of these water molecules will then be attracted and move up or against gravity. So cohesion and adhesion are two really important properties of water molecules necessary for plant growth. Right? Here you see this plant over here is wilting and this one is standing up. Water plays a very important role in plants in turbidity. In what's known as turgor pressure. And when a plant is wilted like this, it's low on water. So you add water to it and it stands right up. And that's because water fills up the cells and provides support for those cells. Finally, in this chapter, we're going to look at acids and bases, so pH. Pure water has the ability to ionize to some extent, meaning release some of its hydrogen ions. And so we have these little positive kind of protons only, and then hydroxyl groups, which are just an OH. So sometimes one of those hydrogen atoms will break off, and then we end up with a hydrogen proton, right? So called a hydrogen ion, or it's just really just a proton, and a hydroxide ion, which is an oxygen with an extra, with only a single hydrogen atom bound to it, covalently as opposed to two. So acids are molecules that release hydrogen ions. So really strong acids will release more hydrogen ions. That solvent or that solution will ionize more frequently, releasing more hydrogen ions than a weak acid. And bases will lower the hydrogen ion concentration, usually by either releasing or adding hydroxyl groups. And so when we add more hydroxyl groups to an acid, that's going to bind and tie up those hydrogen ions, those protons, and decrease the acidity of the solution. So we measure this concentration of hydrogen ions in a scale called the pH scale. And I'm sure many of you are very familiar with it. It's from 0 to 14, and our neutral is 7. So anything with a pH of 7 is considered fairly neutral. The closer to 7 you get, the more neutral the solution will be. As we move down the pH scale, I know I'm moving up here, but the numbers are moving down. So the closer to 0 we get, the more acidic the concentration of hydrogen ions are. And the greater the pH number, so as we move up from 7 to 8 to 9, 10, etc., the more alkaline. So the fewer hydrogen ions we have and the more hydroxyl groups we have. This is because we are looking at the log base 10 of the concentration of hydrogen ions. And this causes us to use these negative numbers. So this is 1 molar. This is 10 to the negative 1 molar. So instead of 6.022, we would be times 10 to the 23. This would be times 10 to the 22, which is fewer. So those numbers get smaller and smaller as our pH scale goes up. And on the right here are just some examples of different compounds that fall within those acidic versus alkaline pH. But pH is really important because those hydrogen ions and even those hydroxyl ions, they have charge. And when you have a molecule or an atom or an ion that has charge, this can really wreak havoc on other molecules in the area, particularly when we're looking in a biological standpoint, when we're looking within a cell. So pH can affect all kinds of different, it can affect how an enzyme works. It affects whether molecules can bind to each other. It affects the rate of a chemical reaction. Sometimes it can speed up a reaction because it's donating charge or energy or ions. Or sometimes it can slow down a charge. Buffers are oftentimes used to maintain a consistent pH so that we can make sure that a solution is stable in order for reactions to occur. And sometimes that the pH can actually affect the ability of things to dissolve in different solvents. So this is where buffers come in, and buffers are really important, mainly because living organisms do not tolerate changes in pH very well. Our cells and most living organisms like things to be at a very constant pH. They don't, if they live in an acidic environment, they need it to always be acidic. Or if they live in a very alkaline environment, they need it to always be alkaline. Our cells prefer things in a fairly neutral system, particularly our bloodstream. And this is because proteins in particular are very strongly affected by changes in pH. Things get too acidic or too alkaline, our proteins can no longer function. And this is because those ions interact with the amine groups in the amino acids of our proteins. So our bloodstream actually has a buffer system in it. It's known as the bicarbonate buffer system. That's what's going on right here. And CO2 will mix with water. We get CO2 when we exhale. CO2 will sometimes mix with water, and we get this H2CO3. This will further break down and give us the HCO3 and the hydrogen ions, and this is known as the bicarbonate buffer system. So here is our bicarbonate, and you can see these arrows are going back and forth, and that's because they maintain things in equilibrium. So that we don't, if we get too much CO2 in the bloodstream, it's going to break it down and form this bicarbonate. But if we get too acidic, then it's going to absorb over here, right? It's going to come back over here and absorb it, and that way our bloodstream does not get too acidic. This acid-base buffer system shifts, and it can either release hydrogen ions or it can absorb hydrogen ions, but the point is that it's going to maintain the pH at a very constant level. The only problem we have with buffers is that they can get used up. So particularly if they are accepting hydrogen ions, then eventually we're going to run out of buffer and we can no longer accept hydrogen ions, and then things will become very acidic, usually very quickly. You may have done a titration in your chemistry lab to show you the using up of a buffer. So that is the end of our chapter 2 on chemical components and structures. These are just some basics that will be part of the foundation for when we start talking about macromolecules and some of the other molecules that make up cells. We will start the next chapter in a few minutes, and let me know if you have any questions. Have a good day.